Okay. Hi, I'm Steffi A. Today we are going to learn about translation. It is from molecular biology. Okay. And what is translation? Translation is the process of protein synthesis. Simply, the translation means it is the protein synthesis. Okay. There are two process in there are two process takes place in the cell for the protein synthesis, and the first step is the transcription. The first step is the transcription. Okay, what is transcription? Do you heard about that? Yes, transcription is the synthesis of mRNA. Okay, synthesis of mRNA from where? From DNA. Yes, from DNA. It's called transcription. And translation, which is the process of protein synthesis, the synthesis of protein, which is the process of protein synthesis. And from where? Yes, it's from mRNA. And we will focus the translation too. Let's watch an interesting thing and we can learn it.
Every cell is like a tiny factory, and at the heart of it is DNA, the instruction manual of life. But DNA doesn't work alone. First, it's transcribed into messenger RNA, a working copy of the genetic code. Then comes translation, where the real magic happens. The mRNA travels to a ribosome, the cell's protein-making machine. Here, each set of 3 bases, called a codon, is read like a word. Enter transfer RNA or tRNA, tiny couriers that bring the correct amino acids, the building blocks of proteins. One by one, amino acids are linked together like beads on a string, forming a protein with a unique shape and function. Just like that, the language of DNA becomes the machine of life, from enzymes and hormones to muscles and skin. Nature doesn't just store data, it with it, and translation is how code becomes creation.
Okay, what is translation? See, the process of decoding of mRNA to synthesize protein is called translation. Okay, the process of decoding of mRNA to synthesize protein is called translation. It takes place in the cytoplasm. Where it occurs, it takes place in the cytoplasm. The process of decoding of mRNA to synthesize protein is called translation, and where it is occur? It's occur in the cytoplasm. Okay, you clear about these 2 points. Okay, just you remember that the translation is decoding of something from someone. From where it is decoding? mRNA. From mRNA it is decoding to an understandable form. What is that understandable form? It is protein form. Okay, decoding of mRNA to protein. Okay, it is called translation. And where does it occur? It is occur in the cytoplasm. Not in nucleoplasm, it is occur in the cytoplasm. Okay, and you know that the transcription is occur in the nucleoplasm, and the translation which means the synthesis of protein from mRNA, it's occur in the cytoplasm. Okay, and what are the requirements? Some requirements are there for the synthesis of protein, and what are the requirements for that process? See, first one, mRNA. Okay, we need to make some dishes, some chicken dishes. Okay, we need first ingredient, chicken is the first ingredient. Okay, and like that, the first requirement for the translation process is mRNA, and where it is occur? It is ribosome. You already learned in cell biology that ribosome is the place for protein synthesis, like a kitchen. Okay, a ribosome is the place of protein synthesis. Okay, and mRNA, and the second one is ribosomes, and the third one is the tRNA which means the transfer RNA. The transfer RNA which brings or it transfers someone transfers something to someone. Okay, transfer RNA which brings something to mRNA or which brings something to the ribosomes. Okay, and the transfer RNA is the third requirement for protein synthesis, and of course we need some amino acids. Okay, amino acids are the building block for proteins. You know that amino acids are the building blocks of protein. Here we are, what we are going to prepare protein. Okay, we need some amino acids for the synthesis of this protein, and here some enzymes are needed for the protein synthesis, and of course we need energy. GTP is energy. Okay, and see what we are learned in this slide. The translation is the decoding of mRNA to synthesize protein, and it takes place in the cytoplasm. And what are the requirements? mRNA, ribosomes, tRNA, of course amino acids, and enzymes and energy. Okay. And what are the key players in translation? What are the key players in translation process? The first one is the mRNA. And mRNA is the messenger RNA. mRNA which means the messenger RNA which has some message, which code for some message. Okay, and it carries a codon sequence. It's a message which brings in a codon form. Okay, the messenger RNA is the which carries a message in a codon sequence. Okay, and the key player in translation is a messenger RNA. Then tRNA. I told about the tRNA which brings someone to somewhere. Okay, the transfer RNA which brings amino acid, which transfer amino acids, which has an anticodon loop. That means it's an anticodon loop which brings amino acid, which carry amino acids to where? Towards mRNA. Okay, and it takes place in the ribosomes. It takes place in the ribosome, the site of protein synthesis, and you know that the ribosome has two units. Yes, large subunit and a small subunits. Okay, and the codon. What is the codon? See, codon is a 3-letter genetic code. You know that the DNA is made up of a double-stranded nucleic acid which has deoxyribose sugar and bases, and which connect with phosphodiester bonds. You know that. And the DNA has these base pairs. Okay, and the DNA get translated to RNA or messenger RNA, and this RNA also has bases. Okay, this RNA also has, RNA is a single stranded form which has bases, and these bases, what are the bases in RNA? Yes, adenine. Adenine pairs with uracil, then cytosine pairs with guanine. Okay, very good. Then the codon is a 3-letter, it's a triplet code, and here we make a codon. You see, here we are going to make a codon. See, A, C, C. This is the 3 base pairs which forms a triplet codon, which code for a single amino acid, and then another codon, you can say that G, U, U, which code for another amino acid. Okay, and another triplet codon C, C, U, which code for another amino acid. Okay, you guys know about the genetic codon in the mRNA. Okay, and here is an example, AUG, which is the start codon, AUG, which is the start codon, which code for the amino acid called methionine. AUG, which is a start codon, which code for the amino acid called methionine. Okay, yes. What are the key players? mRNA, tRNA, ribosomes, and the codon. Okay, what is the first? What is the start codon? AUG, and AUG which codes for methionine. Okay, and the steps in translation process. What are the steps in translation process? There is only 3 steps for the translation process, that is initiation, elongation, and termination. Okay, what are the steps? Initiation, elongation, termination process. Okay, see, the first step is the initiation process. Here you just initiation, that means the starting of a process. Okay, starting of the translation process. Here we need to start something. We need to start a class. We need a teacher, a classroom, and what we need? Students. Okay, like that, the initiation is the starting, and for the start, it have to start a process, or it need some members for some requirements for starting of the process. Okay, and here also the ribosome is needed for starting the process. A ribosome and tRNA and of course mRNA. Okay, and see that the ribosome which binds to mRNA. The ribosome has 2 subunits, large subunit and small subunit, and these 2 subunits which binds to the start codon, of the mRNA, and the initiator tRNA, which has an anticodon, that is AUG is the start codon in the mRNA, which the ribosome bind, and the initiator tRNA, which has the anticodon, that is A pairs with U, and U pairs with, of course, adenine, and G pairs with, of course, C, cytosine, and it has the anticodon UAC, and the initiator tRNA for the AUG, which brings with amino acid, yes, methionine. We learn about this start codon which brings the methionine. Okay, and the tRNA which brings with the methionine, and this methionine comes to the first side of the ribosome, and here forms the initiation complex. The initiation is the process of forming the initiation complex. Okay, here we need the requirements for the initiation complex are ribosomes, mRNA, and the tRNA which brings the methionine. Okay, and the initiation is the initiation complex formation. Simply, initiation is the initiation complex formation which has some requirements: ribosomes, messenger RNA, and the initiator tRNA. And this ribosome which bind to the mRNA and which forms the initiation complex. Okay, and the process of elongation. The second step is the process of elongation. See, the ribosome moves along the mRNA. The mRNA is keeping still, and who is moving? This moving ribosome. The ribosome which moves along the mRNA, and the transfer RNA and the tRNA, this tRNA which brings with the amino acids, this tRNA which brings with the amino acids towards the ribosome, and which always has a, yes, codon, anticodon, connections. Okay, and these amino acids are bring together. These amino acids are linked by peptide bonds. These amino acids are linked by peptide bonds to form a newly bound protein, and it comes out to the ribosome. It comes out from the ribosomes, and the chain grows step by step. The elongation is the process of elongating the protein chain. Okay, the ribosome which moves along the mRNA, the ribosome which moves along the mRNA, and the tRNA which comes with the amino acids, and these amino acids which are linked by peptide bonds in the ribosomes, and which comes out from the ribosomes, and this chain which grows along by step, and this is the elongation process, elongation of the polypeptide chain. Okay, then what is the termination process? When the ribosome reaches the stop codon, when the ribosome which reaches the stop codon, protein gets released. Okay, when the ribosome reaches the stop codon, the protein gets released. And what happened to the ribosomes? And this ribosome disassemble into large subunit and the small subunit. Here you learn about what are the stop codons. Okay, UAA, UAG, UGA. Okay, these three are the stop codons. Okay, UAA, UGA, UAG are the stop codons. Okay, when the ribosome reaches the stop codons in mRNA, this protein gets released from the ribosome, and the ribosome gets disassembled into a large subunit and the small subunit in the cytoplasm. Okay, you learn about the termination process. And what is a genetic code? Some interesting facts about genetic code. See, genetic code is a triplet code. Genetic code is a triplet code, and there are three bases which calls for a single codon, which always call for a single amino acid. Okay, the triplet code is forming with three bases. See, three bases. What is the stop codon? A, U, G. Okay, you just learn start codon is the AUG. Okay, which code for methionine. Okay, this three bases which codes for a single codon and which always code for a single amino acid. And there are 64 codons, and there are 64 codons. The 64 codons always codes with 20 amino acids and 3 stop codons. What are the 3 stop codons? We just learned before. The termination process is UAA, UGA, UAG. Okay, these 3 are the stop codons. There are 3 stop codons. There are 64 codons, and which codes for 20 amino acids and 3 stop codons. Okay, and this genetic code is universal and unambiguous. This genetic code is unambiguous and universal. Okay, some examples are AUG, which always calls for methionine, that is the start codon, and UUU, which is calls for phenylalanine. And some example for the stop codon, yes, UGA, UAG, UGA. Okay, UGA, UAG, UAA. There are 3 stop codons for the, yes. And here we summarize what we learned. The translation is the process of synthesis of protein. RNA into protein, mRNA into protein. Translation is the process of protein synthesis, and in translation which converts mRNA into protein, and it involves, okay, tRNA, then ribosomes, okay, very good, then codons, okay, nice, then amino acid, okay, very good. Then what are the steps? What we learned in steps? Yes, there are 3 steps, okay. Initiation, okay, then elongation, okay, then termination, okay, very good, very good. Then it is the major step for the gene expression, okay. It is a major step for gene expression, okay, then nice. Okay, there are some questions for you. Please answer these questions. Okay, what is the start codon? Yes, can you recollect? What is start codon? Yes, AUG, you said it, yes. Where does translation occur? Where does translation occur? We learned it. Yes, can you recollect it? Where does translation occur? Yes, it is in the cytoplasm. Okay, very good. Then, what does tRNA carry? What does tRNA carry? tRNA carry an anticodon loop, and which brings tRNA, which brings, yes, amino acids. Okay, very good, very good. The tRNA which carry amino acids. Okay, how many stop codons are there? How many stop codons are there? Yes, there are 3 stop codons. Okay, there are 3 stop codons. Yes. Okay, thank you. Do you have any question related to the translation? Yes, you can ask to me.